This method tends to result in integration of fewer copies of the transgene, reducing the risk of multiple insertion and minimizing gene silencing. And minimal damage to host genome. Host genome isn't damaged. Biolistic gene gun method ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ನಾವು ಸೆಲ್ ವಾಲ್ನ ರಪ್ಚರ್ ಮಾಡ್ತೀವಿ ಬೈ ಪಾರ್ಟಿಕಲ್ ಬೊಂಬಾರ್ಡ್ಮೆಂಟ್, ಬಟ್ ಇದರಲ್ಲಿ ಪ್ಲಾಂಟ್ ಡ್ಯಾಮೇಜ್ ಆಗಲ್ಲ. And also plant is not infected because infection causes the genes na ತೆಗೆದುಬಿಟ್ಟಿರುತ್ತೆ. So this is safe, it doesn't damages the host gene. And also plant DNA no hard ಮಾಡಲ್ಲ. Widely applicable, although originally effectively mainly in dicots, ಇದು effectively ವರ್ಕ್ ಆಗಲು ಡೈಕಾಟ್ಸ್ ಆದ್ರೂ ಇವಾಗ ಇನ್ನೂ ಬೇರೆ ಬೇರೆ ಮೆಥಡ್ಸ್ ಕಂಡುಹಿಡಿದಿದ್ದಾರೆ ಮೊನೊಕಾಟ್ಸ್ಗೂ ಈ ಟೆಕ್ನಿಕ್ ಯೂಸ್ ಮಾಡಿದೆ. Then what are the challenges? Limited to certain plant species. ಎಲ್ಲ plant species ಗೂ ಇದೇ ಟೆಕ್ನಿಕ್ ಯೂಸ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. So it is very limited. Then random insertion of tDNA. And tDNA ಪ್ಲಾಂಟ್ ಜೀನೋಮ್ ಅಲ್ಲಿ ಯಾವುದಾದ್ರೂ ಪಾರ್ಟಲ್ಲಿ ಇನ್ಸರ್ಟ್ ಆಗ್ಬೋದು. ಸೊ ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ಅಲ್ಲಿ ಇನ್ಸರ್ಟ್ ಆಗಲ್ಲ. ಸೊ ದಿಸ್ ಈಸ್ ಒನ್ ಚಾಲೆಂಜ್. ಅಂಡ್ ಸೈಜ್ ಲಿಮಿಟೇಷನ್. ದೇರ್ ಆರ್ ಸಮ್ ಸೈಜ್ ಲಿಮಿಟೇಷನ್ ಆನ್ ದ DNA that can be transferred using agrobacterium. The transgene must be of specific base pairs. It cannot be too large or too small. So this is one challenge. Then regulatory and public concerns. Despite its efficiency and widespread use, genetically modified crops developed using agrobacterium mediated transformation face regulatory hurdles and public concerns regarding food safety and environmental impacts. Sometimes ಈ ಟೆಕ್ನಿಕ್ ಯೂಸ್ ಮಾಡಿ ಕನ್ವರ್ಟ್ ಮಾಡಿರುವಂಥ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಪ್ಲಾಂಟ್ಸ್ನ ಕನ್ಸ್ಯೂಮ್ ಮಾಡಿದ್ರೆ ಹ್ಯೂಮನ್ ಹೆಲ್ತಲ್ಲಿ ಪ್ರಾಬ್ಲಮ್ಸ್ ಕಂಡು ಬರಬಹುದು. ಸೊ ಹಾಗಾಗಿ ನಾಟ್ ಆಲ್ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಪ್ಲಾಂಟ್ಸ್ ಕಾಸಸ್ ಪ್ರಾಬ್ಲಮ್ ಇನ್ ಹ್ಯೂಮನ್ ಹೆಲ್ತ್. So this is a concern. So this is one challenge. So next technique is applications of agrobacterium mediated transformation is there. Okay. So what are the applications? Where are the application is used? Pest resistance crops na produce ಮಾಡೋದಕ್ಕೆ ಈ technique use ಮಾಡ್ತಾರೆ. Crops like BT cotton or BT corn has been developed using this method, which incorporates gene of Bacillus thuringiensis to confer resistance to insects and pests. See, so I said about BT cotton and BT corn. So insect resistance gene ana insert ಮಾಡಿದ್ದಾರೆ. How did they insert it? Through this method. Then herbicide tolerant crops such as glyphosate resistant soybean and canola have been engineered using agrobacterium transformation to tolerate broad spectrum herbicides, simplifying weed management. To manage weeds also, the transgene which is resistance to weedicide is also transferred through this method. Improved nutritional content. The development of biofortified crops like golden rice, which has been engineered to produce beta carotene, is an example to the methods used in addressing nutritional deficiency. Then this is disease resistance. Transgenic plants resistance to viral, bacterial and fungal diseases have been developed using this transformation technique, such as virus resistance papaya. So idella examples alle ide, same applications allu barutte. Research tools. So it is a very good tool for research. In addition to its agricultural application, agrobacterium mediated transformation is widely used in basic research to study gene function and regulation in plants. So gene function, gene ಏನು ಕೆಲಸ ಇದನ್ನ ರಿಸರ್ಚ್ ಮಾಡೋದಕ್ಕೂ agrobacterium mediated gene transfer ತುಂಬಾ ಸಹಾಯಕ ಇದೆ. ಕಂಟಿನ್ಯೂ ಮಾಡಿ. So another technique, biolistics. This is also a gene transfer technique used to transfer genes to the plants. What are biolistic? Biolistics, also known as particle bombardment or gene gun method. Particle bombardment ಯಾಕೆ ಹೇಳ್ತಾರೆ? Because